So one of the first things that we need to know when we're working in a microbiology lab is that we're working with the most numerous living organisms on Earth. We're going to be mostly working with bacteria, which are located almost literally everywhere. Now, bacteria far outnumber all life forms on Earth. Not necessarily in diversity, but just in numbers. So, in the large intestine of a human, there's more bacteria than human cells in all our body. Not only that, different kinds of bacteria have been able to conquer extreme environments on Earth, which basically makes Earth being ruled and shaped by bacteria. So you know, the oxygen that we breathe and everything is just the cost of bacteria shaping the world in which we live on. Now, it is estimated that only 3% of bacteria are actually pathogenous or hazardous for men and animals, which are the bacteria that probably take more of the attention in clinical cases, and which are the bacteria that we are trying to avoid. So it's only 3% of all the ones that we know. Now, being very numerous, 3% is a very high chance, 10% as well. So we're going to be careful. Now, we're going to go with hand washing, the effectiveness of hand washing, which is a practice that for us, it seems that has been forever and a very common sense practice. Well, it wasn't that way all the time. There used to be, even in the medical profession, when treating patients, it wasn't a thing that had been done, and it resulted in high mortality until Ignaz Philipp, which is an Austrian doctor, made it part of his routine of washing his hands between patients, and he observed that less and less patients started dying. And that was just because he avoided to contaminate or spread diseases that resulted from microorganisms. So today it became a common practice and a complete scrub to remove every single microorganism, including traction and resting bacteria. It goes about 13 to 14 minutes to complete. Remove both traction and resident microorganisms, which is the first difference that we are going to learn in this class. So we have traction microorganisms, which contaminating microbes, which are easier to remove and basically are not permanent within us, and they can be displaced easily. And then we have resident microorganisms, which are more difficult, and they tend to be present all the time, and it is especially because these have co-evolved with us and they have formed relationships, we with them with us, especially, you know, what the floor gut and all that. So that's this, and hopefully, or more say, all pathogenetics are within the trench of microorganisms. That's something important to know. So what we're going to do is we're going to test the effectiveness of hand washing. We're going to do this with a very simple experiment. So one person in your group is going to contaminate their fingers by rubbing their 2 fingers in the sole of their shoes, and then using a agar plate, which is this plate right here, in which we culture bacteria. Going to rub that in there and then let the bacteria work. The same student on that same hand is going to go wash their hands using my rule to go is a hyperbaric song cycle, performing a proper way to washing your hands, which is washing your hands doing continuous strokes all the way to your wrist. So we can get rid of most, for instance, bacteria that we obtained by rubbing the sole of our shoes. And then we're going to rub those putting fingers on the other side of the plate. The expected result for this experiment is that we want to see whole variety and number of microorganisms in the dirty finger side, and no microorganism or significantly fewer microorganisms of diversity within the clean finger side. Then we're going to test the effectiveness of an antiseptic. The antiseptic that we're going to test, the antimicrobial, is Rocol, which is the detergent that we use to clean our lab benches, and that's that yellow bottles that you see on the bench tops. And we're going to do that by inoculating a split plate with a nose swab. So there are sterile swabs in the material bench in which one student, different from the one that performed the dirty finger experiment, is going to grab a nose swab, perform a nose swab on themselves, and rub that swab in the split plate that I will show you, and then saturate that same swab with Rocol and do a second fill on a cleaner side or a clean side of the experiment. The results we expect are similar. We expect this side to culture some bacteria, so resting bacteria from our same buddy, and in the clean side we expect those bacteria to disappear or significantly decrease. All right, now let's go to the exercise 2. So, what we're going to learn with exercise 1 is that bacteria are present everywhere. We're carrying them